Okay, welcome everybody. Um, Today we're going to talk a little bit about teaching students visual literacy. Um, I use a lot of figures in my teaching, and I've always used figures to explain things to students. But one of the things that I've more recently started to incorporate is teaching students more explicitly the skills needed to interpret the artwork and figures in the textbook. So I want to give you a little bit more information about how I do that and the kind of sources that I've used to develop some activities that I do, some additional activities that I haven't implemented yet, but I think look like good ideas. I want to try to really give you a little bit of a toolkit of activities from today. So I really want to kind of cover three main points today. One is to kind of talk about what we mean by visual literacy. Is it really a skill that can be taught? Why is teaching it important? And then what are some commonly used visual conventions that we can help students better understand? And then by the time you leave with this PowerPoint that you'll have a copy of at the very end, you'll have kind of a whole list of activities that you can start to implement with teaching visual literacy to your students. So, a lot of where this came from, I first got a booklet from the Pearson publisher written by Lori Garrett called The Art of Teaching A&P. And in it, she talks about activities and ways that we can start to be explicit with students about how how to read the artwork, how to read the figures in our course. And then last year, I was also in the book group of Read and Succeed for Reading for Understanding. And there are sections in that book that talk about visual literacy and teaching students how to read figures and kind of making these skills explicit. So, those are two sources for some of the activities that I started to implement in my class. So, Lori Garrett defines visual literacy as kind of the ability to learn from images, basically. The ability to understand and use images, including the ability to learn, which she gets from this reference, which is about teaching visual literacy. And then this is a full PDF that you have access to at this link. This is a study that they did where they actually developed a workbook for students to help students understand some of these artwork conventions that we're going to talk about. Things like how textbooks use color or how textbooks use arrows, those kinds of artwork conventions that you want to teach students how to read. And when they made this workbook for their students and then measured different aspects of student achievement afterwards, the students who worked with the artwork in those workbooks had higher achievement in various measures that they used. So, Lori Garrett uses this as evidence that we really can teach these skills to students and they benefit from us taking the time to point things out to them. So when you start to teach students about figures and how students themselves can read the figures, one of the simplest things that you can do is just show them a figure and ask them, "What do you see?" And sometimes the answer is going to be, "I'm not sure," or "nothing." You're going to be met with silence and a bunch of blank stares. Because for years, I would just explain the figures to my students. I wasn't asking them what they saw in the figure or what they got out of it or what they could understand from it. I just explained to them what was there and then I moved on. When we actually start to ask them what they see, that's a different mental process. And that's a much more active learning process for them to analyze that figure and start to give feedback about what they see. At the beginning, you'll need to facilitate that a little bit. You may ask them, like, what's the title of this figure? What does the caption say? Are there any labels? What color are things? Are there special shapes that you see? And by then you can start to help students to understand that there are certain artwork conventions that they see, and they can use that to help understand what's in the figure. So Lori Garrett outlines a bunch of these these are just common artwork conventions that publishers use. They use arrows, they use color, they can use the coordinate plane, they can show things in a cycle or a sequence, they can use charts and graphs, they can try to set up a figure to show the passage of time. And a lot of times, students don't even understand some of the basic literacy skills, like you can read a figure the same way you read text, which is you can sometimes read from left to right or top to bottom. And a lot of times that's how the artwork is organized, because our eye is, initially drawn to that top left corner to figure out what's happening in the figure. And so when I teach these conventions to students, we start out by talking about how all of these conventions appear in their textbook, because textbook publishers spend a whole lot of money on getting the artwork right, especially in our anatomy and physiology texts, but in other textbooks too. The figures and the artwork that is in your text has kind of really been designed to look that way. I've been in faculty focus groups where they've shown us different figures and said, okay, what do you think about this? And, people are frantically scribbling down everything that the faculty say when they're like, oh, I think this is too complicated for students. Maybe take this piece out, or maybe move this around in some way. The publishers are really interested in that kind of feedback because they want the artwork to be something that faculty use to teach, and they want the artwork to be something that students use to learn. So how can we help students to understand how publishers have devised and designed these figures? And I think one of the steps is actually going through and teaching them about each one of these conventions. So Laurie Garrett first starts with, you have to teach students about arrows. This is a quote from her booklet, and she says, arrows are used so extensively in science images that if a student has a problem interpreting an image, start by looking at the arrows. This is a figure where these red arrows are all denoting magnification, but it doesn't really tell you that anywhere. So they give you a bone, and they've cut this section through and blown it up. Here, this part is blown up. And here they've made another section through and blown it up. And so a lot of times, just by pointing out to students the different ways that the textbook uses arrows, they can really start to interpret figures throughout the whole semester. So I always tell my students, your textbook publisher uses arrows like this to show magnification in this image. They're blowing this up to show you more detail. This is a big, complicated figure that has a lot of arrows, but the arrows are all used the same way. Here, the arrows are denoting the direction of blood flow. Okay, so up here, we're showing kind of a transparent cross-section through the heart. We're showing the flow of blood through the heart. They give you a little bit of a legend here. Arrows indicate the direction of blood flow, and they're using color also. And down here, the arrows are still being used to denote the direction of blood flow. So in these two figures, arrows are being used two different ways. Now, this figure has a bunch of arrows, and they're all being used in different ways. And I always want, this is the way that my textbook represents a few of these homeostatic mechanisms. These are hard figures for students to look at, even if you take the time and slow them down and have them like read the labels and really look through it. So there's these light blue arrows here and here. There's another light blue arrow here, and then there's a red one over here. So these arrows are supposed to denote that this is kind of happening in a loop. Fluid volume will go down, and then it'll come back up. This blue arrow is that it's decreasing, and this red arrow is that it's increasing. And then these arrows are trying to show a sequence of events, right? So when I show my students these kinds of figures, I really have to help them understand how to navigate through these figures. But I've just shown you 3 figures from my textbook where arrows are used differently in each one. So then I present to students other examples of some of these other conventions. So I talked to my students about how information can be color coded and use this figure as an example. And so the blue and purple forms this DNA backbone, and each of these bases has a different color. They blow it up here so you can see the details of how this base pairing works. But then when we have to move back for a little bit wider structural view, the same color conventions are being used. And so I can teach students that they can rely on the same colors to convey the same information. This yellow base is the same as these yellow bases. This green base is the same as these green bases. And they give you a legend to tell you which one is which color. I talk to students a lot about how information can be presented on a coordinate plane. Here, XY axis. Here we're talking about enzyme activity. And so this axis is potential energy, and this is increasing potential energy. And then across the bottom is the progress of the reaction, kind of as time passes, how does the reaction go? We talk about these two different lines. They're using both color as well as this XY coordinate to try to convey the information that more energy is required without a catalyst. In biological systems, that catalyst is an enzyme. And so overall, this graph is showing that activation energy is lowered in the presence of an enzyme. But if you show this figure to a student and say, summarize this figure in one sentence, it takes them a little while to get there. Okay. And in the past, I would always just kind of chicken out and be like, okay, this enzyme shows that this figure shows that activation energy is reduced in the presence of an enzyme. Moving on. And until you actually help students kind of grapple with your images, you skipping over images quickly may lead them to skip over images pretty quickly, right? Like, okay, whatever she said, I'll just keep going. Information can also be presented cyclically or in a cycle. So we talk a little bit about how our textbook presents information as a cycle. Here, this is kind of 3 steps in a cyclical process. The substrate binds to the enzyme. It gets catalyzed into its products, and the enzyme is unchanged and can go on and do this over and over again. Okay, and so we use this as an example to show students that sometimes your book is trying to show you the exact same enzyme. So it does show it 3 different times. This is the same enzyme. It's going through this process. Some textbooks also try to show the passage of time in a stationary figure. So on the right here, these are steps in this process. There's not actually 6 different ribosomes. This ribosome goes through each of these various steps. And so these arrows that are above this mRNA are showing the passage of time as these steps in the process get completed. So Lori Garrett writes in her booklet that she really realized that this is a common misconception that students have when a student said, does ovulation always occur from the right side of the ovary? And she thought that was a hilarious question because these follicles are all throughout the ovary and they're going through various stages of this maturation process. But these 2 figures do indeed show the release of an egg from the right side of the ovary. The student was thinking that this follicle moves through the ovary rather than this being shown a process or a set of steps in sequence. So when you start with an ovarian follicle and it goes through these maturation steps, if you think about how to present that as artwork, we can't draw each of these maturing follicles on top of each other. You won't be able to separate one drawing from the next if you draw those stages one right on top of each other. So instead, we draw those stages right next to each other. But then the student may not pick up on the fact that this is a sequence. This is showing the passage of time in this maturation process. They may physically think that the follicle has to move through the ovary in order for this maturation process to take place. So we can also point out to students that that's a really subtle thing, the idea of steps in a process or the passage of time, and how can your book try to show that in a two-dimensional flat static artwork when really we're trying to visualize it as an actual process. So what are the things that you can do with students to try to help them understand these complicated images that use these different artwork conventions? And one of the things that you can do is really help to emphasize labels with a student. In that earlier example of what do you see, you could also ask students, what is this figure about? And I sometimes do this with a lot of humor. So, for example, this really big, complicated figure, I sometimes ask students, okay, what is this figure going to be about? And they kind of look at me strangely and they're like, it's going to be about homeostasis? Like, yes, very good. And they're like, it can't be that simple. We start pretty simple. Okay, homeostasis of what mechanism? Extracellular fluid volume? Yes, right? And so we sometimes make jokes about it can't be that simple, or is that really what you're looking for, or you just want me to read off that figure? That's a really good place to start when you're looking at really complicated figures. So in addition to talking about what do you see here, you can guide them and facilitate their reading of the labels to try to orient themselves to this figure. Another way that you can emphasize labels is give them a figure with some of the labels blank. You can ask them to try to fill in some of the labels based on what they can see, and that gets them to an analysis of what's in the figure and trying to figure out by kind of backfilling those labels, they can start to see how your textbook uses labels as an art convention. Okay, does anybody have any questions at this point before we kind of move on and start to talk about a lot more specific activities? Does anybody have any questions that you want to unmute and ask or drop in the chat? So, Patricia's excited that I brought up Book Circle and reading for understanding. There's gonna be some more that comes out of Book Talk here in just a second. You'll be very proud of me. Okay, so what I want to model for you is a think aloud with a figure. Okay, so think alouds come from the Reading for Understanding book, but actually both that book and Laurie Garrett's booklet talk about modeling your metacognition when you're looking at a figure for students so that students can understand how you approach those figures and that they can also start to have the same kind of monitoring of their thinking as they're getting confused and clearing up their confusion about figures. And so this is kind of a common thing. You've probably seen this in some PDs that you've been to before. But I just wanted to do this activity really quickly because I also want to show you how I extend that activity a little bit. Also, I'm applying this think aloud to a figure rather than a body of text, which might be something that'll be a little bit different than what you've seen in PDs before too. So I teach biology and my students have had lots of questions about the novel coronavirus since we started. And especially in my general biology class that I taught last fall, my students were asking lots of questions about, does hand washing really work? So I went to look for some figures for a presentation that I did for them in a live class session where we were going to talk about the virus and hand washing. And I came across this figure, and it took me a minute to try to figure out what this figure was trying to show me. It's a hand-drawn figure, and when I actually finally figured it out, I thought, this is the example that I'm always going to use for a think aloud on a visual representation, because it took me a good 5 minutes to try to figure out what this figure was trying to show me. So here we go. Okay, there's a lot going on here. What do I normally do? I normally try to start at the top or at the left-hand side, and they're showing several different things here. So here they're showing soap molecules. They're showing it form a micelle, but here's the virus, and down here they have skin. At first, I thought this line meant this was under the skin, but no, I think this is supposed to be more toward the surface of the skin. This is all kind of happening above the surface of the skin. Here it says virus disintegrating, and there's another virus here that's lower. Okay, so this one starts out intact. Okay, then it started to dawn on me how this figure is actually represented. It is going left to right, but it's kind of flowing from here down and then up. Okay, so here's the intact virus with its membrane. And the soap molecules have a head and a tail that have a little bit different composition. We talk with students about how the head is hydrophilic or really water soluble, and the tail is hydrophobic or very not water soluble, very lipid soluble. And so when you get a bunch together, they form like this. But when there's another membrane around, they start to insert in the membrane and start to rupture it. So I was having a hard time going from here down to here. And then there's more soap. So now this virus is really falling apart. And now we've used soap to disrupt that virus particle, pop open its membrane, and then we rinse all that down the drain, and it's no longer an intact virus. It has disintegrated. Once I looked at this for long enough to figure that out, because at first I was like, I can't show this to students. This doesn't make any sense. And I actually used a different figure with my students. But this became my favorite figure for modeling a think aloud, because it did take me using some of my skills. I had to read the labels. I had to look left to right. And when top to bottom didn't work, I had to kind of go more, like, where is this meandering figure taking me? I think it's a beautiful piece of artwork, and when you figure out what it's showing you, it's a great little representation, but there's a lot going on in this figure. So if any of you guys went to one of my summer workshops, we talked a little bit about how I did a little bit of think aloud with students in a live online class session. And all of my live online class sessions were completely optional for students, and I always sent out a summary for other students. But for the students in the class, we were going to spend an hour really delving into this figure, because it's pretty complicated and it might take them a second to figure out what's going on. This is the type of figure that students are very likely to skip over. This is the type of figure students take one glance at and go, I got nothing, and take off and nope out of looking at what this figure is trying to show them. So I wanted to, in this live class, show them that they could kind of grapple with this figure and figure out what it means and what it's trying to show them. So I started by doing a think aloud on this top portion of the figure. So, well, actually, what we started very first with was a group read that was kind of silent reading, because I didn't assume that students had had a chance to even look at this figure yet. So in my slideshow, I told them, go find this chapter, go to page 677. We're going to look at figure 1910. I gave them about 3 to 4 minutes. And I told them it'll be strange to have silence in a group meeting, but I want you to see just what can you get out of this in the first few minutes. So then when they came back from that 3-minute group read, I had them share, like, how are you approaching this figure? We'd already talked the week before about some of these visual literacy skills. So they were kind of like, I did what you said. I tried to look top to bottom or left to right. I tried to follow the arrows. I tried to read the headings. I'm trying to look at this one piece of information at a time, but it's kind of overwhelming. So then what I decided to do, hang on just a second, I need to take a drink. Sorry, I had a little bit of a cough. So then we decided to delve into this figure in a lot more detail. So I did a think aloud of this top part where we were talking about reading the title of the figure, going through and reading this caption text, trying to talk about these arrows again, meaning magnification. And then I ended that think aloud with, but I wonder what these arrows mean. Okay. And so then I had the students get into breakout rooms and I gave them each a slide to fill out. And their question that they were discussing was, what do these arrows mean? So group one did, what do the red arrows mean? And group two did, what do the magenta arrows mean? And group three did, what do the purple arrows mean? And group four did, what do the blue arrows mean? I had set this PowerPoint up in OneDrive, so I gave them the link to the shared document, and then I put them into breakout rooms, and each group was supposed to discuss what does that color of arrows mean, and I could, and then put their notes from their discussion in that shared PowerPoint file. So I could keep an eye on the PowerPoint file and see how the discussion was coming and go into breakout rooms if I then needed to. But then when we came back, we could all just look through those notes to see the discussion from each of the breakout rooms. It was kind of automatically a way for groups to share out. We talked through kind of a new understanding of this figure as a whole, and students reported. Sorry. Students reported to me that they felt like they had a much better understanding of this figure. The fact that this is a differentiation pathway for cells and that cells are dividing and differentiating through this pathway, not just floating around in this pattern, but it's actually a change that's happening within the cell. And so when I do this fall with my Bio 139 class, I'm also going to then pull in learning catalytics and maybe do some clicker questions with them to actually check for understanding. When we did this, it took up the whole hour, and so I only had just a few minutes at the end to be like, okay, what does everybody think? How do you feel about the figure now? And the feedback that I got from them was that they had a little bit better understanding and they felt better about this big figure, right? And so I was like, it's not just this figure. You can actually use these same skills and techniques that we talked about this whole hour on any of these big giant figures in your class. Because after blood comes heart, and there are several big, giant, complicated figures that they really have to kind of take apart and dissect. So another activity that you can do is about retrieval practice. And I was not aware that we were going to talk about retrieval practice this morning in the keynote. But she's very right. Retrieval practice is one of those evidence-based things that we can do that really show that it helps with learning and it helps with student retention and comprehension. And so one of the ways that I do retrieval practice with my students is I give them lab exams. Sorry, I give them lab quizzes. This is just a lab quiz. This has 10 leader lines on it total. It's going to be worth 10 points. But I often have to explain to students that they can't just stare at a figure and that's memorizing it, right? They can't hope to just stare at a figure and they'll automatically remember all of the parts and how to spell them. So when I first started doing lab quizzes, I was like, well, this is this is slammed. I tell them this is figure 34.1 in your lab book, and next week you'll have a quiz on figure 34.1. So study figure 34.1. And I would come to unlock the lab, and they'd be in the hallway with their book staring at the figure, and they'd come into lab and they'd still be staring at the figure, and they'd even be like, I'd be passing it out and I'd be like, close your book, and they'd be like, okay, and slam their book shut and start to write on it. And they could get a few, maybe half, maybe 3/4, but they're like, oh God, I can't remember what that other one was. Because their entire learning process with that figure was very passive, it was just staring at the figure. So what I do now is I incorporate different retrieval practice activities when I'm going to do this lab exam for students, especially in person. So I'll say, next week you'll have a quiz. It'll be 34.1. Go ahead and get that out. Take a few minutes to look at it. Maybe I'll give them a practice quiz so they'll see just by staring at it, you can only get about half of them right. Then I have them cover the labels in the book with sticky notes or paper and practice writing down and practice trying to get close enough in spelling to all of the various structures that they're looking at. And so once I've had them go through that practice and then they take the lab quiz, they're much more likely to be able to remember all 10 structures and write those down than if they were just staring at the figure. So I emphasize to them that these are the kind of active ways that they want to interact with figures to attempt to learn it rather than just staring at it and hoping that they'll remember. Another thing that you can have students do is draw and trace. And it doesn't even have to be good. It's just the activity of close examination of the figure that you're trying to reproduce. So I traced this. I put this piece of paper over the figure and tried to trace it. And it's okay. It's not that great. But this is an example of one of the things that I have my students do. They can stare at this figure all day long, but they get so much more out of it when they actually are required to trace and label their tracing accurately or somewhat accurately. Asking a student to draw what they visualize when they read text is another way that you can have them, especially if they're reading words and they're trying to visualize a process or they're trying to visualize a passage that they're reading, having them draw what they're imagining is another way to interact with the material. And it doesn't have to be perfect, and it doesn't even have to be good, and it honestly doesn't even have to look like anything. Like, this is supposed to be part of the skull, and I just like drew that circle. It was like, it doesn't have to actually be representative of anything. It's the process that you're going through that's helping you look at the details of the artwork. What another activity that I do with students is I ask them leading questions about the artwork. So one of the things that Lori Garrett talks about is when you think that students are when you think that students are not getting what they need out of a figure, you can try to facilitate their interaction with that figure to try to point out the parts of that that contain the most information or the information that they're supposed to be getting. So here's a figure that I sometimes give to students on a worksheet, and then here are a set of questions that I ask. And the first one is, if you look at this graph, what is this a graph of? And again, this is just encouraging them to really look at the labels. And some of them are like, it can't be that simple. You really just want me to write this down and write this down? Yes. And then I ask them to really look at these labels: optimal temperature for a typical human enzyme, optimal temperature for a heat-tolerant bacteria enzyme. And so to get these temperatures, all they really need to do is know how to use this graph. This will be about 37 or 38, which is normal human body temperature. This one will be about 78 or 80. And then kind of lead them in a little bit of analysis. Why are they different? So one of the things that I have put in the files in the team, in the channel associated with the PD team in Teams, are 3 examples of some worksheets that I've put together that have lots of these kinds of activities. And you can see that I've picked some difficult figures for students to really try to piece out what's happening. And then in the lecture room, I would have them work in groups to discuss the answers to the questions. And the questions are designed to really have them look at things like the labels. It may ask them, what do the colors represent? What do the arrows mean? Where do the arrows representing here? Some of those conventions that you want them to pay attention to. Now, Lori Garrett says that if you want students to appreciate the art, you should use the art. And a couple of different ways that she recommends is to include images on quizzes or exams, or to use the figure in clicker questions or polling. So in one of my classes, I use Poll Everywhere. Poll Everywhere allows you to upload a figure. And so I use this figure in this poll question and say, click on the dipeptide. So here's a single amino acid, and here's a single amino acid, and they've been joined together by this peptide bond to make a dipeptide. And so after having discussed that with students, if I'm kind of trying to do a measure of understanding, then I can give them this poll question and kind of see where people are clicking on this diagram. Are people kind of getting the idea of which is the dipeptide? Learning Catalytics also allows you to upload a picture and have students click on a region of the picture. So this is click on the area of the figure that shows venous valves closed due to contraction of skeletal muscle. And so here's the vein, and here's the muscle relaxed, and here's the muscle contracted. And they give a box and an arrow to show this more magnified. This is how my book shows magnification. This one's still open, but this one is closed. And so I can mark this region, because I don't know if they're going to click the smaller or the magnified, but I can make this whole region be the correct answer. So if they click here or here, they get the point for clicking on the closed valve. So one of the other resources that you have that's going to be in files is the preface to Lori Garrett's booklet, The Art of Teaching A&P. And it's a really good summary of why we should teach and work with students on visual literacy. In her booklet, this is another activity that she has. And she talks about assigning students some kind of figure or a table or a chart to prepare on their own. And then they come to a group activity where they have to apply that activity or to that artwork to their question. She talks about providing a case study or a medical problem where they've prepared by looking at the artwork. So now they can apply what they've learned from that figure to the question that they've been given. Some additional things that I'm thinking about trying to implement that I haven't actually implemented yet. One of the things that she recommends is maybe if you have some sort of a pie chart in your textbook, have students convert that to a table or a different kind of graph. So when she presents this figure, she talks to students about how there's a lot of percents here. We're going to work with percents today. And here is a line that show that although these two aspects of this artwork are related, there's a separation between these percents over here and these percents over here. And so she'll focus with them on this left side that has this pie graph, and she'll actually have them do kind of a think, pair, share, where they begin with some independent work, the think part, where they try to put this pie graph into a table. And then she has them pair up and talk about how they're approaching it. Because when you're trying to put this in tabular form, you're going to want to do probably organic versus inorganic, and then add in all these examples of the inorganic components. So once they start to talk to each other, then they can kind of expand how they're looking at converting this to a table. And then she has students share out their discussion while she actually tries to make a table in the front. And that she says that students really start to appreciate the information that's in a pie graph when they really start to wrestle with other ways to represent that same information. One of the things that I want to try to do with students is to ask them about editing figures. When we ask students kind of which figures are more confusing, which ones did you want to skip over, can you tell me which ones I need to help facilitate with you in more detail about? One of the things that we can also ask is, how would you try to make this figure more understandable? Where was it really confusing? And if you were an art editor, how would you try to clear up that confusion? How would you change the colors? How would you add labels? How would you space it out? How would you try to represent that this is a process that can repeat and recur in a cycle, or any of those other types of conventions? And then one suggestion that's in both places is talking about comparing two different representations of the same information. So you can kind of give students two examples of a structural representation and kind of ask them what's similar and what's different about these two. You may get some superficial things in the beginning. What's different? Well, they're different colors. Okay, what else is different? And further facilitation until they actually understand how the figures are representing the same information, and it will help them with that visual representation of the structure that they're trying to study. So in Microsoft Teams, we're all in a PD Day team. We are in this Jefferson Faculty PD Day 2021 under Teaching Your Students Visual Literacy. Under the files are all of those examples, and I'm getting ready to upload my PowerPoint so that you'll have access to it. So we have about 7 minutes left. I was trying to leave at least 5 to 10. So what questions do you have, or what types of visuals do you use with students when you're doing your teaching? I apologize for the coughing earlier. I like inhaled something out of the air that wasn't compatible with me.
Valerie, it's Stacy. Can you hear me?
Yes, I can.
So my writing students have to do scholarly research. They have to include so many peer-reviewed articles published in scholarly whatever. You're with me. The types of figures in there are most often mathematical ones. So they are percentages and medians and numbers, lots and lots and lots of numbers. And without some sort of basic math literacy, they really struggle with trying to turn those into sentences. And I absolutely refuse to take just lists of statistics and facts in papers, right? They have to analyze that. So I think talking about number literacy is just as important as talking about visual literacy in terms of reading data and charts, if that makes sense.
It absolutely does make sense. And she does talk about some computational literacy and graphical literacy as specific types of visual literacy that you can teach to students. She has a whole section in here, and like if you contact one of the Pearson reps, you can get this little booklet sent to you, and it's super cool. But she talks about graphical literacy, and graphical literacy is the ability to understand quantitative information such as data, and that a lot of our students don't come with that, even to science classes don't always come with that kind of background, but especially to writing classes are not going to have the um, a lot of the specific training to be able to interpret those. One of the things that she talks about is that activity of like working with pie charts, and so this having an activity where the student themselves manipulates some of that data. So how would you take this data and maybe represent it in a different way? So that's a thing that would probably need to be facilitated with them. Like that's not an activity that you can just send them off on their own to figure out. She talks a lot about helping students to understand labels and axes on graphs. And she says that's one of the reasons why in our laboratory classes that we sometimes have students collect data and graph data so that they can figure out that they can't turn in a graph to you that says line 1 and line 2. They have to tell you what line 1 and line 2 are. That they can't send you where the title is, urine experiment. It's like, no, you're looking at the amount of a substance per volume of urine. And so it is something that I think is really difficult. By teaching students some of the basics, some of the reading labels, reading the captions, reading the titles, and really trying to figure out why the artwork was designed in the way it was, to get to look that way and to represent it that way. I think they can start to get at the pieces of that, but it's extremely difficult if they don't have any computational background or graphing or analysis of that in their history, right? In what they've already been exposed to.
I'm certainly nodding my head at you, even though you can't see me since my camera's not on. But I love the idea of teaching them to represent the data in multiple forms. If they can represent it differently, then they've shown that they've understood it, and then they can move to the analysis form. So I really like that, and I'm going to have to think about that some more and how I can do that.
One of her practical examples is that we always give students a table about blood types and their percentages in the population. So her first activity is take that table and make a graph out of it. And then her second activity is the one about take the pie chart and turn that back into a data table, so that you can kind of see that you need to represent your data tabularly to get it into these various forms. And that by working with or manipulating a set of data they're already provided with, then they can start to see how data manipulate, why the data looks the way it does in a published paper.
One other thing, and then I promise I'll shut up, but you were talking about labels, and of course teaching people to read labels and legends on maps and all of those kinds of things, right? You can't understand a visual graphic if you don't have the key to it. We know that. They don't always know that. But one of the things I've noticed lately, I've been doing some sustainability reading because of this course I'm going to teach starting in September. I'll tell you about later. But anyway, so I've been really looking at energy, and there are all these abbreviations on these tables that I don't have a clue what they mean. And the labels and keys and legends don't tell me. I have to read the article before I can figure out, like I have to match all those little pieces together to try to even figure out what some of those abbreviations mean.
You got to go back to the first mention of it too, because a lot of times they will only tell you once and then just use the abbreviation the whole. And so sometimes it's in the abstract, and sometimes it's in the introduction, and sometimes it's in the methods. And
yeah, and I think that's something I need to really talk to them about this year that I haven't before. I've talked about reading the labels and reading the intro and the abstract, and then you can skip through and look at the tables. Well, you may not be prepared for those tables.
So yeah, so one of the things when I am actually presenting this to students, oh, I'm not gonna be able to find it really quickly. One of the things that I talk to students about is there's a figure in my book that relies on labeling that was in the previous figure. It uses P for proton and N for neutron, and it just assumes that you got that from the previous page, the previous figure. And so in the PowerPoint that I present to students, I've made a visual literacy video for my students to watch. And I point out to them that sometimes what you're looking for isn't in the figure. You can do all of your skills. You can look for the legend, you can look at the colors, but there's some symbols that were defined earlier or in a prior chapter, and they think that just because you already talked about this thing that you may still have that same understanding of the terminology, so that so I tell students they may have to back up a little bit to um, be able to figure out what the figure's trying to tell them. Well designed artwork doesn't make you do that, but poorly designed artwork can make you have to go back and search for what does this even mean. Patricia put in the chat, one student my questions respond to when they analyze graphics is why that type of graphic or visual was the best one for the author to use for their purpose. And I think that that's a good sign. I think that that's a good question too. Like, what, out of all the money that the publisher could have spent to make this figure look this way, why did they do this? Out of all the time that the author spent writing this journal article, why did they represent their data in this way? And maybe they did have some other choices, or maybe it was just the nature of the data lends itself. But those are all really good questions to have students think about. Right. And in my own teaching, like I said, I had to shift from just explaining every figure myself to putting some of the onus on them in a safe space and in a safe way to start to grapple with it and ask and answer questions where they're actually working with a figure, to emphasize to them that the artwork is important and it's something that you may have to sit down and grapple with. But that's where a lot of the meat of the explaining is, once you figure out what all the parts are trying to teach you. Yeah, you can, like I said, I've made my students a whole video, and it's optional. I don't make them watch it, but I tell them I think it'll help them. And I do some of these activities in live class sessions, because that's kind of a good place to take one thing that was in one of my videos that everybody watched and kind of do it in a little bit more detail. So, well, thank you guys very much for being here. If anybody has any more questions or comments that you want to leave or drop in the chat, you're welcome to. But I also know that we need a little bit of a break before everybody moves on to their 2:00 appointment. Katie, I agree. There could be a whole class devoted to understanding graphics. And it's, that's one of the reasons why I've just kind of wanted to put it into some of the general, like the study skills and the literacy skills that I try to impart on my students. Those are the things that they're going to take out of my class that they're always going to really remember, probably even more than the content. But that's fine, because if they can read figures that they come across on a regular basis, then I think they'll be able to pick up, whatever they don't remember or need additionally. So, yay.